Okay, so this chapter is nutrition and metabolism. So this is actually two chapters in the textbook that we've kind of combined. They're both really short. And I'm telling you that because there's a lot of overlap in the two, and I just kind of put them all in there. So if you get to a slide later and it looks repetitive and you're like, she already said this exact same thing in another slide, that's why. It's not anything new or tricky or I just kind of did one and then did the other. So there is some overlap in the two chapters. So this is going to be the last chapter that's covered on exam 3. We've got respiratory, digestive, and then nutrition and metabolism. Okay, so this talks about the essential nutrients. These are nutrients that are, or substances that we have to have in order to survive. So you've got your carbohydrates, your lipids, your proteins, minerals, vitamins, and water. So minerals, vitamins, and water, and then most of your biomolecules. When I say biomolecules, I'm talking about proteins, carbohydrates, lipids, and then nucleic acids, which you'll notice is not on here. So all of your biomolecules except for your nucleic acids. Carbohydrate molecules are just sugar molecules. So if you have a monosaccharide, it's just a single sugar molecule. Examples of monosaccharides are glucose, fructose, and galactose. If you were to look up the structure, you would see it's just one simple single sugar molecule. Disaccharides are if you have two sugar molecules connected. Sucrose, maltose, and lactose are examples of those. And then if you're talking about a polysaccharide, it's a long chain of sugar molecules connected. The main examples of those are going to be starch and cellulose. Carbohydrate digestion is essentially breaking the bonds in between the long chains of polysaccharides or in between the two sugars involved in a disaccharide until you get a single sugar molecule. And the majority of carbohydrate digestion is going to take place in the oral cavity and then in the small intestine. So it's going to start in the oral cavity, and we kind of talked on that with the digestive system. It starts with that enzyme salivary amylase. So salivary amylase is synthesized and released from the salivary glands, and it breaks the chemical bonds in between the glucose molecules and starch. The extent to which it can break it down depends on how long the salivary amylase is allowed to interact with the starch. So if you're taking your time and chewing slowly, then you're going to get a lot more starch digestion in the oral cavity than if you were to just eat really rapidly. Okay, so once you create that bolus of food and then you swallow it and it goes into the stomach, the low pH of the stomach activates the salivary amylase. So salivary amylase, it's the enzyme that breaks down carbohydrates or breaks down starch. It is synthesized and released in the oral cavity. That's where it does its job. Once it's been swallowed with the food bolus and it hits that acidic environment of the stomach, salivary amylase is done doing its job. So then it will spend some time in the stomach and then we'll move on to the small intestine. This is where we're going to resume the breakdown of carbohydrates, and we're going to do that using the enzyme pancreatic amylase to further digest the carbohydrates. So it's similar to salivary amylase, but it's coming from the pancreas. It's one of those digestive enzymes from the digestive juices. Okay, dextrinase, glucomylase, and maltase are all things that we call brush border enzymes. These are enzymes that are embedded in the epithelial lining of the small intestine, and they break the bonds of oligosaccharides and disaccharides to yield individual sugar molecules. So if we're talking about a disaccharide, it would look something like this. It's just two sugar molecules with one bond connecting them. So one of these brush border enzymes, either dextrinase, glucomylase, or maltase, would come in and break that bond and create two individual sugar molecules. Alternatively, if you have an oligosaccharide, it would look something like this. It's not a super duper long chain, but it's also a little bit more than a 2 or 3 carbon sugar. In that case, the brush border enzymes would come in and essentially break the bond here, break the bond here, break the bond here so that you're releasing individual glucose molecules as you go. And then I've got the names of these specific enzymes. Lactase is an enzyme specific for lactose, and sucrase is an enzyme specific for sucrose. So you can see down here in the box, here's a partially digested starch, partially because it started in the oral cavity. Then you have your pancreatic amylase, which will continue to break it down until you get to these oligosaccharides, although you could, depending on how big it was when it came in, you could, you might get individual sugar molecules, you might get disaccharides, or more likely you're going to get the oligosaccharides. Then your brush border enzymes pick up, and that's when it does what I just described, where you'll end up with individual glucose molecules. So for the absorption of carbohydrates, the monosaccharides that get released whenever we digest those oligosaccharides, you're going to get glucose, fructose, and galactose. Those are going to get absorbed across the small intestine epithelial lining, and then they're going to go into the bloodstream. So if you look at this picture here, this is going to be like inside the small intestine. The different color circles are going to be glucose, fructose, and galactose. And then they're just going to kind of absorb across that epithelial lining, and you see the blood is right there, and it will get picked up into the blood. Now that blood is going to go straight to the liver. So remember, all the venous blood from the small intestine is going to go through that hepatic portal vein. We're going to take it to the liver first. In the liver, the fructose and galactose are going to get converted into glucose. So we need glucose. So some of that glucose gets absorbed from the GI tract. If we absorbed fructose and galactose, it's no big deal. It's going to go straight to the liver first, and the liver will convert it into glucose, so that by the time we get it to the inferior vena cava, we're talking about all glucose. Okay, so from there, any of the glucose that can be taken up from any body cell in the bloodstream, we can store it in the liver as glycogen, or we can convert it into triglycerides and store it as adipose tissue. Cellulose is a carbohydrate component of cell walls, plant cell walls. It is not chemically digested because we don't have the enzymes to do it. So cellulose is essentially non-digestible, so it serves as kind of like a fiber because it will stay in the GI tract, and it kind of adds content to the lumen, and then that facilitates water moving into it, and that facilitates then movement of the GI tract. Okay, so cellulose and starch are very similar. They are both long chains of glucose. But the bond that links the individual glucose molecules together is different in cellulose and starch. So for starch, we do have the enzyme to break it down, salivary amylase, pancreatic amylase. Cellulose, because the bond is a little bit different, we don't have enzymes that can do it. So it essentially stays in the GI tract. It serves as like a fiber. Anytime you have a fiber in the GI tract, it pulls water in, and that helps to facilitate the movement of the contents. Okay, protein digestions. Proteins are polymers, which are, again, long chains of amino acids. So when we're talking about digestions of proteins, we are talking about the release of the individual amino acids so that our body can then take those and then use them to build up new proteins that we need. So anytime we're digesting these things, we've got to break them down into their smallest unit component in order to actually absorb them. If they stay disaccharides in the case of carbohydrates, or dipeptides in the case of proteins, where we haven't broken it down to either a single sugar or a single amino acid, we're not going to get absorption. So we've got to digest them down to those individual units. So in this case, it's individual amino acids. All the enzymes that digest proteins are released as inactive enzymes. We talked about that in the digestion chapter, and they have to be activated. That is because we have proteins all in our cells. And if we release them in their active form, it's just going to start degrading all of the proteins in our cells, and it's going to be very bad for our cells. So we release them as these inactive proteins or inactive enzymes, and then when they get where they need to go, where that area can handle them, then they get cleaved into an active form, and from there they can start doing their job. So the one we talked about in the digestive system was pepsinogen, and it got activated or converted into pepsin by the hydrochloric acid in the stomach, and then pepsin started to break down the proteins. Okay, so protein digestion is going to happen mostly within the stomach and the small intestine. Okay, protein breakdown in the stomach. The protein hydrolysis begins in the lumen of the stomach with the enzyme pepsin. So again, we saw this in the digestive chapter. Pepsin is created in its inactive form, which is pepsinogen, and then it gets released by those chief cells. And then if you remember, the hydrochloric acid that was released from the parietal cells caused the low pH. The HCl also helped to denature or unfold the proteins, but then HCl causes the conversion of pepsinogen into pepsin. So it basically turns on the enzyme or activates it, and then pepsin can then start to break down the protein. So you can see in this little experiment here, if you put the protein in the hydrochloric acid, not a lot happens. Basically right here you're denaturing the protein. And then if you put it in just pepsin by itself, you're going to get some denaturation, but because the protein hasn't really been denatured, we're just cleaving away at the bonds it can find. But if you have both pepsin and acid, you've got everything you need to denature the protein and cleave away at it, and you can see you get complete digestion of the protein in this one. So from the stomach, then it's going to move into the small intestine. The high pH of the small intestine is going to inhibit the pepsin. So just like when we looked at salivary amylase, it was active at a regular pH, and when it hit the stomach, it was inactive. Pepsin is the opposite. Pepsin is active in the acidic environment of the stomach, and once it gets to the more alkaline or more neutral environment of the small intestine, it doesn't work anymore. 3 of the enzymes that continue digestion for protein are synthesized and released from the pancreas into the small intestine, again in their inactive form. So these names are going to get kind of interesting. Trypsinogen, chymotrypsinogen, and procarboxypeptidase. So these are all inactive forms of enzymes that are going to be involved in digesting proteins. They are synthesized and released from the pancreas, and they go into the small intestine. So about the time that your chyme from the stomach is moving into the small intestine, these enzymes are going to move from the pancreas into the small intestine and meet them there. Okay, once these inactive enzymes get into the small intestine, you've got an enzyme enteropeptidase. This enzyme is synthesized by the small intestine and released into the lumen, and it's going to lead to their activation. So it actually activates trypsinogen into trypsin, and then trypsin goes on to activate the other 2. But enteropeptidase is an enzyme that is made and released in the small intestine, and it is used to lead to the activation of all of the other inactive enzymes that came from the pancreas. Okay, we also have brush border enzymes for protein breakdown, and they are responsible for breaking that final bond between 2 amino acids and a dipeptide. So dipeptide, we're just talking about 2 amino acids connected. And so once we break that final bond, we have 2 individual amino acids, and they can be absorbed. The brush border enzyme aminopeptidase generates free amino acids from the amino end of peptides. So let me kind of say all that again. Dipeptidase is useful when you have a dipeptide. So you have 2 amino acids that are linked together, and the dipeptidase will come in and just chop that one bond in between the 2 of them. So if you have amino acid here, bond here, dipeptidase basically just cleaves it right there. If I have a peptide, I'm a terrible drawer, especially when I'm trying to use a mouse, so forgive me. This is what we're going to call a peptide. Aminopeptidase would cleave here, and then this guy would be free. And then it's going to cleave here, and then this guy would be free. At that point, you would have a dipeptide, so dipeptidase would come in and cleave that one. But so aminopeptidase just kind of plucks one off the end to generate individual amino acids. Okay, so once we have all of the free individual amino acids, they get absorbed across the small intestine epithelial lining and they get into the bloodstream, just like we saw with the individual carbohydrate molecules for the sugars. And then from there, just like for carbohydrates, they're going to go to the liver. There's no conversion that needs to happen there. But then eventually they're going to get out into our systemic system, and all of our cells have access to these amino acids, which serve as individual building blocks so that we can rebuild the proteins that we need. Alternatively, they can get converted into glucose by gluconeogenesis in the liver, kidney, if that's what our body needs. Okay, so I would spend some time looking at this because even though it may look daunting initially, it's very helpful to visually see all of that stuff that I was just talking about. Okay, so what you have here is this is your partially digested protein molecule coming into the small intestine from the stomach. Right here is where you're going to get the enzymes and digestive enzymes that are coming from the pancreas, and they're coming into the small intestine. So you can see everything kind of meets up right here. So in that digestive juice, you had trypsinogen, chymotrypsinogen, and procarboxypeptidase. These are, again, the enzymes that are going to degrade proteins, but in their inactive form. So this dark blue means inactive. Once it becomes light blue, it's an active enzyme. So brush border enzyme, enteropeptidase, will activate trypsin. It converts trypsinogen into trypsin. Now trypsin can activate the other two. Trypsin converts chymotrypsinogen into chymotrypsin, and procarboxypeptidase into carboxypeptidase. Okay, so now you have three, oops, I messed that up, active enzymes to break down these partially digested proteins. And so what you're going to end up with is these right here, peptides. So that's when your other brush border enzymes are going to come into play, peptidase and dipeptidase, and basically start chopping these down until you get individual amino acids. The individual amino acids will then cross over the epithelial lining of the small intestine and go into the blood capillary. And then from there it's going to go to the liver. From there it would go to the inferior vena cava, back to the heart, and now we're pumping out these nutrient molecules to all of the cells of our body. Lipid digestion is a little bit different than the others because lipids are so variable in their structure. It's not as simple as a long chain of this or a long chain of that. They arrange their building blocks so much differently. So we're really going to talk about cholesterol and triglycerides. So one thing that they all have in common, though, is that they're lipophilic. They're not water soluble. We remember that from, the chapter on the endocrine. So triglycerides are the ones I always say look like the letter E. It's got a glycerol backbone, and then it's got 3 fatty acid chains that hang off of it. And essentially to digest So this would be a triglyceride that I just drew. Essentially, to digest that, you basically have to break the bonds between the fatty acid molecule and the glycerol. And then there's cholesterol, and cholesterol actually doesn't require digestion. We can absorb that molecule as is, so that's good. Okay, so lingual lipase is an interesting enzyme. It's produced by the salivary glands in the mouth. So it's part of saliva, but it doesn't actually do its job there. It just kind of hangs out. The optimal pH for lingual lipase to work is between 4 or 5. So it doesn't actually become activated and do its job until it reaches the stomach. It's just kind of comes from the mouth, it's along for the ride until it gets to the stomach, and then that acidic environment activates the lingual lipase, and then it starts to do its job. So once it's made its way to the stomach, the triglycerides will undergo partial digestion by the lingual lipase that just came in from the mouth and got activated in the stomach, and then gastric lipase. We saw gastric lipase. I think I talked about it. Maybe I didn't, but it was produced by the chief cells in the gastric pits. Okay, so we've got lingual lipase and gastric lipase. They start to digest the lipids to roughly 30% of the triglycerides to diglycerides. So a little bit of digestion. The majority of it's going to happen later in the small intestine. So we're going to move that lipid from the stomach into the small intestine, and that's where the pancreatic lipase is going to be the main enzyme that comes in from the pancreas and is responsible for breaking the remainder of the lipids down. So pancreatic lipase breaks the triglyceride into a monoglyceride and 2 free fatty acids. So what's going to happen is here's your glycerol backbone, and here's your fatty acids. What we need for lipid digestion is you're going to end up with your glycerol backbone, and you've got one remaining fatty acid attached to it, and then here's your other fatty acid, and here's your other fatty acid. So we basically pulled this one and this one off, and that's what we consider digestion of lipids. Okay, so because the lipids are not soluble in the aqueous environment of the lumen of the small intestine, because they're lipid soluble, remember, we need to first emulsify the fat so that our digestive enzymes, our pancreatic lipase, can get better access to it. So when I say emulsify, I mean basically break it down into smaller droplets. So it's just like putting oil in a bucket of water. All the oil is going to come together and form one large droplet. And if you can think about an enzyme now trying to break that down, it only has access to that outer layer of the droplet. So you're not going to get really efficient breakdown. So if we go in and emulsify this or break it down into a bunch of tiny droplets, then our enzymes have much better access to the fat to start breaking it down. So the way we're going to do that is by bile salts, which is a component of the bile that's going to get released from the gallbladder. So in the digestive section, I talked about bile being important for fat metabolism, and here you can see how it's working. So the bile is going to come in and essentially break that large lipid droplet up. You can see that here, one large lipid droplet, and then we're going to turn that into a bunch of smaller lipid droplets. And then our pancreatic lipase can start really breaking it up. So the bile salts are amphipathic molecules. If you don't remember what that means they have a polar part and a non-polar part. So all of the polar parts are going to kind of unite together and be in contact with the aqueous environment of the small intestine, and then the non-polar parts are going to be kind of on the inside, and that's going to create this structure called a micelle. So you might look those up if you, I know we've talked about them, at least 2401 covered them. I can't remember if we've talked about them in 2402, but if you don't remember what a micelle is, go back and look that up, and that's what the bile salts are creating from our fat. So the pancreatic lipase starts to digest the lipids inside the micelles, And again, we're breaking it down into what you see here, pulling those two free fatty acids off. Also inside the micelle, you're going to have cholesterol and any lipid-soluble vitamins that got ingested with the meal. We're not digesting those, we're not breaking them up, but they're going to hang out with the lipids because they got to get absorbed together. So we don't have any brush border enzymes that are required for the lipid digestion. It's all this pancreatic lipase, in addition to the gastric and lingual lipase that we saw earlier. The digested triglyceride, which again consists of the monoglyceride and the two free fatty acids, is going to enter the epithelial cell of the small intestine. So I'm going to talk about this and then I'll kind of show it on the next slide. The bile salts stay in the small intestine lumen where they're going to get recycled and reused. Inside the epithelial cell, the individual fatty acid will reattach to the monoglyceride to form the triglyceride. So originally it went into the cell like this with our glycerol and one attached fatty acid and your other two fatty acids. Once it's in the cell, we're going to reattach it to reform the triglyceride. So we really only digest it long enough to get it into that small intestine epithelial cell. Then we're going to wrap it in a protein coating to form what's called a chylomicron. So we wrap it in a protein coating, and then the chylomicrons get packaged into these secretory vesicles. So now we're putting a bunch of these into a larger secretory vesicle. So you can see that we're kind of building this larger structure within that small epithelial cell. So the chylomicron is too large at that point to get into the capillaries, so that's why instead it goes through the lacteals of the lymphatic system. So when we talked about the lymphatic system, at least in the lab, I know I mentioned that the lacteals you find in the villi of the small intestine and that they are used to absorb lipids, and this is what's going on there. Because of the way that we digest and then we reform our lipids and then we wrap them in this protein coating, we've got this large structure that simply isn't going to get into the capillary. So instead we send it into the lacteal, which goes into the lymphatic system, and we know that the lymphatic system will eventually dump back into the circulatory system. So I say the same thing I said about the proteins. I would spend a lot of time just really looking at this and going through step by step and making sure you understand the individual steps. I think this is a really good picture to kind of show it in a stepwise manner. So here you've got lipid droplets coming in, partially digested, roughly 30% from the stomach, using our lingual lipase and gastric lipase. Again, this is where we're going to get digestive enzymes coming in from the pancreas. It's going to meet up in the small intestine. And then you've got bile salts. So then we're going to take this large lipid droplet, we're going to break it down based on the bile salts into these smaller lipid droplets and forming these micelle structures. Pancreatic lipase is going to act on them inside the micelle, and this is what's going to happen. You got your triglyceride, and it's going to break it down into a monoglyceride and then your two free fatty acids. So the bile salts will stay in the lumen of the small intestine. We're going to recycle those and reuse those. And then the triglycerides and the two free fatty acids will come back into the epithelial cell. You can see that they're going to get reformed into the triglyceride there. Wrap it in a protein coat called a chylomicron. So this is what they look like there. And then package all those into a vesicle. And at that point, of course, it's too big to go into the capillary, so it's going to go into the lacteal, which will eventually make its way back into the blood. For nucleic acid digestion, nucleic acids are polymers or long chains of nucleotides. We've got the two types of nucleic acid polymers are going to be either DNA or RNA. This is going to be in any kind of plant or animal that you eat. A nucleotide monomer is made up of 3 different parts. You've got sugar, phosphate group, and nitrogenous base. So we need to break down the long chain of nucleic acids into individual nucleotides, which this whole structure right here is nucleotide. And then further, we're going to break the bonds between the component parts for complete digestion. The enzyme deoxyribonuclease breaks down DNA, and ribonuclease breaks down RNA into their individual nucleotides. And then we've got brush border enzymes phosphatase, that's going to break this bond, and nucleosidase, that's going to break that bond, so that we get all of the component parts. And then all of the individual parts, the phosphates, the sugars, and the nitrogenous bases, are going to get absorbed across the epithelial layer into the bloodstream. Celiac disease is an autoimmune disorder that affects about 1% of the population, and it's basically when gluten, which is a protein that you find in wheat and rye, stimulates an autoimmune response where it attacks the villi of the small intestine. So in the digestive system, I talked about the function of the villi and how they're there to increase the surface area so that you get maximum absorption. So if those start to get damaged, it interferes with your nutrient absorption. So common foods that have gluten, breads, pizzas, pastas, things like that, it causes abdominal pain, chronic diarrhea, and then eventually you get nutrient deficiencies because you're essentially attacking the villi of the small intestines. So there's not really a treatment for this, but if you avoid gluten, if you follow a gluten-free diet, then you can avoid that autoimmune attack and you can work through it that way. So this is where that kind of one chapter end and the other chapter started. We've kind of combined it all into one unit. So this is where if you may start to see some repetition of things that we've covered already in this unit. Okay, so nutrients include most biomolecules, vitamins, minerals, and water. They're obtained through the diet. We process them by the digestive system. Nutrition is the study of the means by which living organisms acquire the nutrients that they need to grow and sustain life. The nutrient biomolecules that are carbohydrates, lipids, and proteins. And then again, nucleic acids are not considered nutrients because they're not required for survival. Vitamins are organic molecules required for normal metabolism, but they're only present in very small amounts in foods. We categorize our vitamins in one of 2 different ways, either water-soluble vitamins versus fat-soluble vitamins, or essential versus non-essential. So water-soluble vitamins dissolve in water. That's going to be the B vitamins and vitamin C. They're easily absorbed into the blood from the GI tract. And for water-soluble vitamins, if you have an excess of what your body needs, it just excretes it with the urine. So you're not going to overdose on water-soluble vitamins. Fat-soluble vitamins, on the other hand, dissolve in fat, not in water. That's going to be vitamins A, D, E, and K. They get absorbed from the GI tract within those lipid micelles when we eat a fatty meal, and then ultimately go into the lymphatic capillaries. If the dietary intake of fat-soluble vitamins exceeds what we actually need, then the excess gets stored in the fat. And if you continue to consume more than we need and you store more and more, it actually can reach toxic levels, and they call that hypervitaminosis. Functions of the fat-soluble vitamins. Vitamin A is known as retinol. This is a precursor molecule for the formation of a visual pigment called retinal. Vitamin D, calciferol, is modified to form calcitriol, which is a hormone that increases calcium absorption from the GI tract. Vitamin E is tocopherol, helps to stabilize and prevent damage to cellular membranes. And then vitamin K is a vitamin that we use for synthesis of certain blood clotting proteins. So this is a table that shows all the vitamins. I'm not requiring you to like memorize this. I put this in here because people do tend to be interested in this more than some of the other topics. So once everybody is done with this course and you've got a chance to breathe and you want to go back, it's just some good information on the different vitamins. Minerals are inorganic ions. So where vitamins were these larger organic molecules, minerals are just simple inorganic ions. Calcium, sodium, potassium, iodine, zinc, magnesium, and phosphorus, those are all examples of minerals. They all come from the diet. We've got a daily requirement for all of our minerals. And then that amount that we are required falls into one of two categories. Major minerals are minerals that we need at levels greater than 100 milligrams per day. That's going to be calcium, chlorine, cobalt, magnesium, manganese, phosphorus, potassium, sodium, and sulfur. Yes, I would probably know which ones are major and which ones are minor. Trace minerals are those that we need at less than 100 milligrams per day, and that's going to be chromium, copper, fluorine, iodine, iron, selenium, and zinc. Same thing as the vitamin chart. This is a mineral chart. I'm not going to require that you memorize this for the exam, but it's in there for your interest for later. This clinical view talks about iron deficiency. I'm really only concerned that you guys kind of focus on this section right here. So basically, iron deficiency is one of the most common nutritional deficiencies that we have. It's going to present with fatigue, weakness, pale skin, headaches, sensitivity to cold temperatures. And you can basically become iron deficient if you're not consuming enough or if you're losing excess iron somewhere else. So how much we consume is going to be based on the diet. Heme iron, which is iron that comes from meat, basically, is going to be more readily absorbed than non-heme iron. Non-heme iron is the iron that comes from iron-rich plants. Also, absorption can be altered by other things that we eat at the same time. So vitamin C can actually increase the absorption of iron if they're consumed together, but something like tea actually can interfere with iron absorption. So how much iron we actually take in is dependent on the source of the iron and what else is in the GI tract at the same time. And then if you have things like Crohn's disease or celiac or something like that, where you've got changes to the intestinal lining, that can also affect how much iron actually makes it into your system. The major carbohydrate that we use for energy is glucose. We take it in through the diet, and then if we take in one of the other monosaccharides, galactose or fructose, those go through the hepatic portal system. They go to the liver where the hepatocytes are going to convert those two back into glucose, so we're going to get more glucose for our system. Next, just some definitions on different types of carbohydrates. So starch is a polysaccharide. It's a long chain of glucose molecules. You'll find starches in things like potatoes, breads, rice, and grains. Lactose is a disaccharide, which is a combination of glucose and galactose that you find in milk. Sucrose is a disaccharide containing both glucose and fructose that you find in table sugar, maple syrup, and fruits. Again, cellulose is that plant polymer. It's glucose bonded together, very similar to starch, but the bonds are in such a way that we don't have enzymes to break those bonds like we do the ones in starch, so we can't digest cellulose. So the consequence of that is that cellulose remains in the GI tract, acts like a fiber, pulls fluid into the lumen, and then facilitates the movement of the contents through the GI tract. More on lipids, you've got the triglycerides. These are Another major energy providing molecule obtained in the diets is fat. So carbohydrates are more of your short term, and triglycerides are more of your long term in terms of energy providing molecules. So butter, animal fat, milk, cheese, oils, these are going to be high in triglycerides. The absorption of fat-soluble vitamins requires the absorption of fats, because those fat-soluble vitamins have to come in those mice cells with the fats in there, with the triglycerides in there. So you'll need to, for maximum absorption of fat-soluble vitamins, you should have some fat being absorbed as well. Cholesterol is another type of lipid component of animal-based products, so meat, eggs, and milk, because it's within the plasma membrane of those cells. Plant cell plasma membranes do not contain cholesterol. We also produce cholesterol through metabolic pathways in the liver. So the liver synthesizes cholesterol. So we can either get cholesterol through our diet, or we can create it in the liver if we're not taking enough. If we are taking enough in the diet, our liver's not going to go on overdrive trying to make it. One important thing about cholesterol is it's a precursor molecule for the formation of steroid hormones, estrogen, progesterone, testosterone, bile salts, and also fat-soluble vitamins. Adequate dietary protein gives us the amino acids that we need to build and replace the proteins that our body needs to use. So we can get protein from meat, dairy products, poultry, fish, beans, and nuts. Just some other definitions. Vegetarian is an individual who does not eat meat, fish, or poultry. Lacto-ovo-vegetarian, an individual who does not eat animal flesh but does consume milk, eggs, and cheese. Individuals who do not eat any animal products are called vegans, and then pescatarians are individuals who don't eat most animal flesh but will eat fish. So we all know that animal products are rich in proteins, rich in amino acids. Plant-based proteins are often incomplete as an individual source. However, if you eat a variety of the plant-based proteins, then you can get all of your amino acids that way. So if you decided, I'm going to be a vegetarian, but I'm only going to eat pinto beans and nuts, then you're going to end up lacking amino acids because those only would have certain ones in them. But if you eat a wide variety of plant protein sources, it'll be just fine, and you'll get everything that you need. One thing about proteins is they also serve as an important source of nitrogen. Nitrogen is important for synthesizing DNA and RNA. So that's another kind of key role that proteins have in our diet. This just talks about absorptive and post-absorptive states. Basically, after you eat a meal, for the next 4 hours, your body is going to be constantly absorbing those nutrients that you took in. After about 4 hours, if you don't have another meal, you kind of stop doing that. But keep in mind, all the while that you're bringing in these nutrients, you can't just obviously let the levels rise in your blood. So like, for example, with our glucose, we can't just let it keep going up and up and up, but rather we have these hormones, insulin, in that case, to kind of move it out of the blood and into the cells of our body so we don't get out of kind of homeostatic range. A little bit of the anatomy of the liver. So the functional unit of the liver is a lobule. So these were the ones that I compared to the bronchopulmonary segments of the lungs. So functional unit of the liver is a lobule. The cells of the liver are called hepatocytes. So within the lobule, you've got these cords of hepatocytes. These just liver cells kind of lined up end to end. You've got oxygenated blood that's coming in through the hepatic artery. Here, I don't know if y'all can see that arrow. You've got deoxygenated but nutrient-rich blood that's coming from the GI tract into the hepatic portal vein. That's going to run through, it's going to go through the sinusoids that are in with the hepatocytes. It's basically just going to run this direction, and then eventually it will flow out through the hepatic veins into the inferior vena cava. You also have bile canaliculi that are going throughout because the hepatocytes are making bile that we're going to then send down to the gallbladder. So I mentioned that cholesterol can either be taken in through the diet or it can be synthesized by the liver. If it's going to be synthesized in the liver, it starts with a process called beta oxidation, where the fatty acids are converted into acetyl-CoA. Acetyl-CoA is then used to actually make the cholesterol molecules. Each person synthesizes cholesterol at a different rate, inversely related to how much they're taking in through their diet. So that just kind of makes sense. The more you're taking in through your diet, the less your liver is going to synthesize. And if you're not taking in much through your diet, then your liver will synthesize more. After the liver synthesizes the cholesterol, it is going to either release it as what's called a very low density lipoprotein, or VLDL, or it can use it to synthesize into bile salts and release into the small intestine. So we have to transport our lipids in a special way because they're hydrophobic, so they don't like just free floating throughout the blood. It's just like our lipophilic or cholesterol-based, steroid-based hormones. So we first wrap them in a protein coating, and then we transport them that way. So the protein coating kind of protects them from the aqueous environment of the blood. The term lipoprotein refers to this protein-wrapped lipid structure. So the chylomicron that we discussed when we talk about lipid digestion is an example of a lipoprotein. So we have got different kinds of lipoproteins. We've got very low density lipoproteins, or the VLDLs. We've got LDLs, which is low density lipoproteins, and HDLs, which is high density lipoproteins. And you can see that the difference is basically the composition of lipids that's within each one. So you've got a large triglyceride, well, this is a regular chylomicron, a large amount of triglyceride and a chylomicron and just a little bit of cholesterol. And then you can see that the level of cholesterol, and when we're talking about VLDL to LDL to HDL, the level of cholesterol goes up from 14% in a VLDL to 45% in an LDL, and then we're back down to 20% in an HDL. So I don't need you to know those numbers by any means, but just kind of understand that it's the changing composition of what's in the chylomicron, or what's in the lipoprotein, that determines whether it's a VLDL, an LDL, or an HDL. Okay, so what's really important about the difference between those three transport molecules, I'll get to in just a second. Okay, so when we're transporting the LDL molecules from the liver to the peripheral tissue, it's going to start out as a very low density lipoprotein and low density lipoproteins. And so those are going to be the molecules that move the cholesterol from the liver to the tissues of the body. So this is your hepatocyte here. It has just synthesized the cholesterol. It's going to package it in a VLDL, and it's going to leave, and that is now traveling to deliver the cholesterol to the different cells and tissues of the body. So it's delivering here triglyceride. So triglyceride gets released, and that's going into the adipose connective tissue. So as we reduce the amount of triglyceride that's present in that vesicle, it goes from being a VLDL to an LDL. The LDLs have receptors on certain cells. You can see that here. So the LDL will come onto a cell that has a receptor in it, and it gets endocytosed. And so the remainder of that will then be in the cell. So that's different from the HDLs because the HDLs are going the opposite direction. HDLs are associated with the transport of lipid from the peripheral tissues back to the liver. And that's why we kind of think of them as the good cholesterol, because they're kind of pulling lipids out of our tissues and then taking them back to the liver, as opposed to just constantly delivering lipids like the VLDL and the LDLs do. So the proteins for these are formed in the liver, but they go out into the blood essentially empty. They don't have any lipids in them. So again, here's your hepatocyte. And an empty HDL. So it's basically like an empty vesicle that then leaves the liver and goes into the blood. And as it's traveling through the blood, it will pick up lipids. So here, it's picking up triglycerides and putting it into the HDL molecule. And then once it's full, we're coming back over here, once it's full, it will deliver that back to the hepatocyte. So LDL and VLDL take lipids and cholesterol from the liver to the peripheral tissues. HDL does the opposite. It brings lipids and cholesterol from the peripheral tissue back to the hepatocyte. And that's mainly what I want you to know about the difference between those molecules. So we can test our cholesterol levels with a blood draw. They're going to test your cholesterol, they're going to test your LDL, they're going to test your HDLs. And basically, any one of those readings is good, but it's really kind of collective pool of information, because what they're looking for is a healthy ratio of HDL to LDL, because that's going to give you a better estimate of, the overall processing of your lipids and cholesterols. So again, HDLs are considered the good cholesterols because they're transporting the lipids, which cholesterol and triglyceride, from the arterial walls and other peripheral tissue back to the liver for elimination, as opposed to the LDLs and VLDLs, which are doing the other. They're actually making lipid deposits in the peripheral tissue, starting from the liver.